Welcome to an in-depth exploration of the cutting-edge science behind age reversal and cellular rejuvenation. Recent breakthroughs suggest we may be able to reverse the biological clock at the cellular level, opening unprecedented possibilities for human health and longevity. Before we discuss reversal, we need to understand what aging actually is at the molecular level. Aging is fundamentally the progressive accumulation of damage to our cells, tissues, and organs over decades. As we age, our cellular function progressively declines, shown here as decreasing capacity from youth to old age. Scientists have identified 9 hallmarks of aging, including telomere attrition, genomic instability, and most importantly for our discussion, epigenetic alterations. A major breakthrough came with the discovery that aging leaves predictable marks on our DNA through epigenetic modifications. DNA methylation patterns change so predictably with age that they can be used as a molecular clock to determine biological age. Methyl groups attach to DNA at specific sites, and the pattern of these attachments changes systematically as we age, creating an epigenetic clock. In 2013, Steve Horvath created the first highly accurate epigenetic clock by analyzing methylation at 353 specific DNA sites. The Horvath clock can predict chronological age with an accuracy of plus or minus 3.6 years across all tissues and ages. This raises the crucial question that drives modern aging research. If epigenetic changes mark aging, can we erase these marks and reverse the process? The answer came from an unexpected source. Research on stem cells led to the discovery of 4 proteins that can literally turn back cellular time. Japanese scientist Shinya Yamanaka made a Nobel Prize-winning discovery. 4 specific proteins could reprogram adult cells back into stem cells. These 4 factors, abbreviated as OSKM, are October 4, SOX2, KLF4, and MYC, and they work together to reset cellular identity. The Yamanaka factors act like a molecular reset button, transforming old, specialized cells back into youthful, pluripotent stem cells. Initially, this was developed to create patient-specific stem cells for tissue repair and drug testing, not for aging research. The challenge was that full reprogramming erases cell identity completely, which would be dangerous for living organisms. If you fully reprogram all cells in an organ, they lose their specialized function. A liver cell must stay a liver cell while becoming younger. Partial reprogramming applies the Yamanaka factors briefly, just long enough to reset the epigenetic age without erasing what type of cell it is. Research shows that applying the factors for less than 2 weeks can reset age markers while maintaining cell type and function. This breakthrough means we can make cells younger without turning them into stem cells, opening the door to whole organism rejuvenation. In 2016, Juan Carlos Ispizua Belmonte's lab published a groundbreaking study showing age reversal in living mice. The team used mice with progeria, a genetic disease causing rapid aging, as their experimental model. The results were dramatic. Treated mice showed improved function across multiple organs, including heart, kidney, and pancreas. Most remarkably, the treated progeria mice lived 30% longer than untreated controls. Critically, the treatment did not cause cancer or tumors, which was a major safety concern given that MYC is an oncogene. Let's examine what actually happens inside cells during partial reprogramming at the molecular level. Partial reprogramming reverses several hallmarks of aging at once, not just one pathway. Each cycle of treatment can reduce biological age by 1.5 to 3.5 years, according to epigenetic clocks. The treatment affects DNA repair, mitochondrial function, telomere length, protein homeostasis, and stem cell function simultaneously. Cellular energy production improves dramatically, with mitochondria showing 20 to 40% better function. Interestingly, the treatment can reverse cellular senescence, making zombie cells functional again instead of just killing them. One of the most remarkable demonstrations came from restoring vision in aged and damaged eyes. In a 2020 Nature paper, the Sinclair lab at Harvard showed they could restore vision in mice with glaucoma by applying partial reprogramming to retinal cells. The optic nerve, which had been damaged, regenerated, and visual acuity, improved significantly in the treated animals. Nerve regeneration rates increased more than 5-fold compared to untreated controls. This is crucial because it shows that partial reprogramming doesn't just slow aging, but can actually repair accumulated damage. A critical question arises. If we're making cells younger and more proliferative, why don't they become cancerous? The answer lies in the brief, pulsed application. Short exposures reset age without disrupting the tumor suppressor networks that prevent cancer. The natural question is whether these findings can translate to humans. Let's examine the current state of clinical development. Multiple companies, including Altos Labs, Calico, and Retro Biosciences, are now working on translating this research to humans. The field is progressing through standard drug development stages, from preclinical studies through early human trials. Initial human applications will likely focus on localized treatments for conditions like macular degeneration and joint degeneration. Realistic estimates suggest first approved therapies could reach patients within 5 to 10 years if trials succeed. A major technical challenge is how to safely and effectively deliver reprogramming factors to human tissues. Researchers are exploring 3 primary delivery methods, each with distinct advantages and challenges. Viral vectors offer efficient gene delivery, mRNA provides temporary expression like COVID vaccines, and direct proteins are safest but hardest to deliver. Adeno-associated viruses are currently the most popular method because they're relatively safe and work well in animals. The success of mRNA vaccines has inspired research into using similar technology for temporary expression of reprogramming factors. Let's examine the quantitative models that describe how aging progresses and can potentially be reversed. Biological age is a function of chronological age, environmental exposures, and genetic factors. The rate of aging can be modeled as the accumulation rate minus the repair rate. In typical aging, damage accumulates faster than our bodies can repair it, leading to net aging over time. This graph shows normal aging as a straight line, while treatment creates periodic dips where biological age is temporarily reduced. The total rejuvenation effect is the integral of repair rate minus aging rate over the treatment period. An important question is whether there's a theoretical limit to how much we can reverse aging. Based on current data, each treatment cycle appears capable of reversing 2 to 4 years of biological age. Let's dive deeper into the molecular mechanisms by which Yamanaka factors reset the epigenetic landscape. The Yamanaka factors function primarily by reorganizing chromatin, the complex of DNA and proteins that determines gene accessibility. The OSKM factors recruit enzymes like TET proteins that remove methyl groups from DNA, erasing age-associated methylation patterns. TET enzymes convert 5-methylcytosine to 5-hydroxymethylcytosine, which is then removed, returning the DNA to its unmethylated youthful state. Beyond DNA methylation, histone proteins are modified back to youthful configurations, opening chromatin for gene expression. Different species age at vastly different rates, and understanding why helps us optimize interventions. Maximum lifespan varies enormously across species, from 2 years for mice to 200 years for bowhead whales. Lifespan roughly scales with body mass to the 0.2 power times metabolic rate to the negative 0.3 power. Naked mole rats are remarkable for maintaining youthful physiology for decades and having extremely low cancer rates. A critical question is whether the treatment will scale effectively from mice to humans given our longer lifespans. The answer is complex. Humans have more accumulated damage, but our slower aging may allow for gentler, more sustained interventions. To know if treatments work in humans, we need reliable biomarkers that change faster than waiting for lifespan effects. Epigenetic clocks remain the most validated biomarkers, providing age estimates that correlate with mortality risk. Multiple biomarker types exist: DNA methylation, inflammatory markers, phenotypic age, proteomics, and metabolomics. Phenotypic age combines clinical chemistry measurements into a single biological age estimate. The most reliable approach combines multiple independent biomarkers to create composite aging scores. Any intervention that manipulates cellular identity raises legitimate safety concerns that must be addressed. The biggest fear is that making cells more youthful could make them more prone to cancerous transformation. Safety protocols involve careful dose escalation with intensive monitoring to detect any signs of unwanted cell proliferation. Another concern is that cells might lose their specialized function if reprogramming goes too far. Risk is proportional to dose times duration divided by monitoring frequency, suggesting careful titration is essential. To minimize risk, treatments use pulsed intermittent dosing rather than continuous exposure, and target specific tissues rather than whole-body treatment. If age reversal becomes reality, the societal implications extend far beyond medicine. Preventing age-related diseases could reduce healthcare spending by trillions of dollars over coming decades. Estimates suggest age-related disease burden could decrease by 60% or more with effective interventions. People could remain productive workers and contributors to society for additional decades. Critical ethical questions arise about who will have access to these therapies and how to ensure equitable distribution. A common concern is whether extending human lifespan would worsen overpopulation problems. Early treatments may extend health span, keeping people healthy longer, without necessarily extending maximum lifespan dramatically. The field is rapidly evolving with several promising directions for future research and development. Research is focusing on identifying key age-related pathways that can be targeted for therapeutic intervention. Personalized medicine approaches are being explored to tailor treatments to individual needs and genetic profiles. The role of the microbiome in aging and age-related diseases is an area of increasing interest and research. The potential for age reversal in other species, such as dogs and cats, is also being explored for its implications on human health and longevity. As research progresses, we can expect significant advancements in our understanding of aging and age-related diseases, leading to the development of innovative therapeutic strategies and interventions.